Our speaker is Whit Athey, who was the editor of the Journal of Genealogy 2005 to 2009, and also 2011 to 2012. He is the administrator of 5 Y chromosome projects. He's the author of the popular web-based program Haplogroup Predictor, which provides Y haplogroup predictions from Y STR values. He's a member of the ISOG committee. That's, correct me, that's the International Society of Genetic Genealogy.
Yes.
Member of the ISOG committee for the Y phylogenetic tree. He's a retired biophysicist whose working career was primarily at the Food and Drug Administration, where he was the chief of one of the medical device labs. He received his doctorate in physics and biochemistry at Tufts University and undergraduate engineering and master's math degrees at Auburn University. For several years during the 1980s, he also taught one course each semester in the electrical engineering department of the University of Maryland. Besides his interest in the application of genetics and genealogy and anthropology, he is an amateur astronomer and has his own small observatory near his home in Brookville, Maryland. So I'm going to turn this over to Whit. Go again with screen sharing. Hopefully it'll continue to work.
Okay, I'll do that in just a moment. I should have edited that information a little bit more. I no longer live in Brookville and I no longer have my observatory. I live in Baltimore now.
Okay.
So,
but I'm sure you can still see the stars from Baltimore.
Occasionally.
Whit.
Joel, are you seeing the first slide okay?
Yes.
Okay. All right. We're going to be talking today about primarily autosomal DNA. That's the DNA that you have in your 22 sets of chromosomes. We'll briefly mention Y-DNA and mitochondrial DNA, but primarily this is about autosomal DNA and what you can do with it. Joel, my presentation isn't advancing. Oh, now it is. Okay. Well, first of all, what is DNA? Let's just make sure one is on the same page with some nomenclature. DNA is an acronym for deoxyribonucleic acid. It's just a very, very long molecule, a chain of subunits. And there are only 4 different subunits in this long chain. The subunits are called bases, and their names, the bases have chemical names, but we just abbreviate them to the first letter of each one. We refer to them as A, C, G, and T. And you're probably familiar with the double helix structure of DNA, showing how the bases are paired up along the double helix. Now, how do you specify a piece of DNA? Well, it can be completely specified by its sequence. Here's an example of a sequence, and that's really about all that you can say about it. Just what are the letters along the strand? If you could put the DNA from all 22 chromosomes together and stretch it out, it would be 3 billion bases long and about 6 feet long. And you have 2 copies. So really you have about 6 billion bases of DNA. And this 6 feet of DNA is coiled up inside a box that's about 10 or 20 microns on the side. So you can tell that it's quite an engineering feat. You can imagine unwinding all of that DNA, making a copy of it, and then separating it out into two new copies when cells divide. It's really quite incredible. Here's a diagram of the 22 sets of autosomal chromosomes. Of course, you have a 23rd pair, either an X and a Y or two X's, depending on if you're male or female. Now, the chromosomes don't look like this sort of X shape, except just before they divide, these are about to separate into two copies.
I can't see the pointer.
Well, can you see the pointer that on the screen? We checked that earlier, but
Yes, we can.
okay, great. Thank you. Now, in transferring DNA from parent to child, each parent, of course, has two sets of chromosomes, two copies, one that they got from each of their parents. Now, the child cannot receive two sets of chromosomes from each parent. So each parent can transmit just one set. But how do they decide what they're going to pass on? How is the half that they can transmit selected? One possibility would just be to pick one chromosome out of each pair of the 22 and pass that along. The number of combinations that you could get in terms of possible children would be about 4 million. But evolution apparently wasn't satisfied with that. So the actual process is more involved, and the process is called recombination. It's mixing and matching of the two chromosomes in each pair, and then picking one chromosome out of that mix and match process. And with that process, there are trillions of possibilities in children. Now, I'm going to illustrate this by actually my own chromosome 4. I've got my parents' chromosome 4 diagrammed at the top. The light blue my father got from his father, the pink my father got from his mother, and it also my mother's parents contributed the dark blue and the green. So what my father actually passed to me was from his father up till the point 137 million bases along the chromosome, and then the rest of it, the pink shaded, came from his mother. Now, on my mother's side, she did some more recombination, and that's generally true. You'll find that females do more recombination than males do. So this shows the mosaic of that she passed along to me, made up of her parents' two chromosome 4s. So how does this process work? Well, the parents must start with a germline cell, which has a full set of 22 chromosome pairs. Sort of counterintuitively, the first step is to double the number of copies to 4 sets. But then the cell divides, but just before dividing, you get this mixing and matching. And finally, 4 cells end up with one set of chromosomes. In this next little video, a cartoon video, this process is illustrated. Excuse me. In this video, just 2 chromosomes are illustrated, a large one and a small one, rather than all 22. The because it would just be too confusing. It's sort of confusing enough as it is because I can't figure out how to slow it down enough. But let's take a look at it. Finally, we end up with 4 cells that could be these could be egg cells or they could be sperm cells. And you see the DNA that ends up in these 4 different cells is all different.
Wow, this is like too much.
Here we have another illustration of how DNA is passed down the generations. If you look up at the top line, the great grandparent generation, we have two autosomal, I'm sorry, 1 chromosome pair out of the 22. Then the smaller chromosome is the Y chromosome. It doesn't come in pairs. And finally, the little circular one is mitochondrial DNA. For the Y chromosome and the mitochondrial DNA, we know exactly where that came from in terms of, say, our great grandparents. I have Y chromosome. I got it from my father. He got his from his father. He got his from his father, and right up an all male line with very few changes. They're passed down. Similarly, the mitochondrial DNA comes down a strictly female path. Both males and females have mitochondrial DNA, but males don't pass it on. But now, look what happens to these larger chromosomes that represent the autosomal chromosomes. They get mixed and matched in each generation as you come down, so that finally, in the present generation, you have quite a mosaic from all 8 of your great grandparents. So if you have your autosomal DNA tested, what you get is the DNA base at about 500,000 locations sprinkled around your whole genome. But you get 2 results at each location because you have 2 copies of every chromosome. And there is nothing in the testing results alone that'll tell you which base came from which parent. So like one line of your raw data might look like this: chromosome 12, the location along chromosome 12, and what bases were there. In this case, C and T. And the report is just done in alphabetical order. We know whether the C came from the father or the mother. Now, what is a match? What you get from the testing companies is a whole list of matches with other people, but what does a match mean exactly? Consider DNA from 2 different people, the same stretch of same location. We can see that, could you get me some water? We can see that the top line, this sequence, is not like either of the ones in the other person. But the second line, the second sequence, is the same as the paternal line in the other person. And so we would say that these 2 people are half identical on this segment of DNA because they match on one of their chromosomes and not the other. Now, in this particular example, the paternal sequence is exactly the same as the paternal sequence in the second person, and similarly for the maternal ones. In this case, we would say these two people are fully identical. This doesn't usually happen except between siblings. Now, here's a case where two people do not match. Their sequences are not the same on either of the two chromosomes. So we would say are not identical, but we could only say that because we're looking at the sequences and we have them separated according to the paternal side and maternal side. But if a computer is looking at these sequences and going from location to location, the computer would end up saying that these two are half identical because it matches. Looking at the maternal sequence, you can see that it matches down below on the paternal side till you get to this point where there's a C, but down here there's a T. But the first person has a T at the other chromosome, and so the computer would say, "Oh, these still match." And in fact, along this whole stretch, the computer would say, these are half identical, even though they're not identical. So how can you tell a pseudo match like this from a real half identical match? And the way that you have to do this is by looking at a very large stretch of DNA. If you should have the case where at a certain location, you have the same base in one person, like the two T's shown in red, and the other person has two C's, well, you know for sure that they are not identical in this region. But that's the only way you can tell the difference. So what you need is a very long stretch of half identical or fully identical comparisons. So how long does the stretch have to be? Well, it depends on how sure you want to be that the match you're having reported to you is real and not a pseudo match or accidental match. So roughly you need a stretch of about 700 locations that you're comparing. I should have mentioned earlier that the testing companies test a location about every 3,000 or 4,000 locations. They're not testing every single location. They're only testing those that are known to vary in the human population. About 99% of the human genome is identical in everyone. And so we're only looking at the locations where there is variability. Was there a question? Anyway, you need a stretch of about 700 SNPs where you have a match, say a half identical match, and that usually covers about 5 million bases of the sequence, of the fundamental sequence. There's a measure of this that's called centimorgans, and it's abbreviated just CM. So we would like to see at least 7 centimorgans, that the match has at least 7 centimorgans of matching bases. Usually you'll get a report from the company of your matches, and it will tell you how long each match is. And it usually will be in centimorgans, although some companies give you the percentage of DNA that you share. So here's an example from one of the companies. This is from 23andMe, actually. And this is a comparison of my genome to a second cousin of mine. And you can see that we have half identical matches on 5 different chromosomes. You also get this same information in tabular form. And this, you'll notice in these two columns, you get the centimorgan of the match and the number of SNPs that are involved. And as I said, in order for to really be considered a real match, you want the number of SNPs to be, say, 700 or more, and the number of centimorgans to be at least 7. So this is an excellent match. All of these are actually, and they're all half identical matches. Remember, you won't really see any identical matches except between siblings. Now, there's a bit of variation in the number of centimorgans of matching DNA between different types of relatives. Like this orange shaded histogram here shows you the distribution of centimorgan matches between first cousins, and the brown is between siblings. As you get further and further out in your relationship to other people, other cousins, you can see that the distribution starts to overlap considerably. And so if you have a match with someone else of 100 centimorgans, the best guess of the testing company might be you're a third cousin to this person, but you could be a fifth, might be a fourth, or you don't really know because there's so much variation involved. Now I want to turn to the main point of the presentation. How do you determine how recombination occurred in a family? And there are two main approaches. One is a sort of computational approach, and one is a more graphical visual approach. And I'm going to discuss the visual approach first. The other one is probably going to take more time than we have available to us today, but I will provide a link to an article that I've submitted for publication, which will describe both approaches in detail. So you don't really need to be taking notes, if anyone is, during this presentation, because you'll have a copy of the slides at the end, and you'll have a copy of an article, and you'll also have some other tools that I'll make available. First of all, why do you want to do this? Why do you want to have, why do you want to know how recombination happened? Well, it's useful to have a complete map of your chromosomes in a family of siblings, showing where each piece of each chromosome came from in terms of the parents, for example. This next slide is going to show my own chromosome 10 and that of my sister and my two brothers. Each of us got slightly different versions of our parents' DNA. For example, I got mostly on this particular chromosome 10, I got most of my DNA from my father's mother and a little bit from my father's father. And it's more evenly distributed among my mother's parents. But my sister got almost nothing from my paternal grandfather. My first brother got almost nothing, also almost nothing from the paternal grandfather, but nearly everything from the paternal grandmother. My youngest brother got more of a mix. Now, down at the bottom is a match that showed up database. This person had a match to 3 of us, but not the 4th. And if you look as to what part of these chromosomes this person matched, he matched me, my sister, and my youngest brother. in this region. So what you see is that the thing that is in common to the three of us is the green DNA. And so because of that, we would know that this person is related to us through my maternal grandmother. And that's the value in having diagrams like this. When you have a match to someone else in the database, you know immediately through which grandparent you are related to this person, even if you never heard of them before. And that's just the result. We now know that that's the green DNA. The advantage to this is it can narrow your genealogical focus and concentrate just on the line that you know you have to be related to this other person to. But how can you determine the diagram that we just saw? Where did I get that? Let's go back just a moment. Note that where there's a change in the DNA, like from the maternal grandfather to the maternal grandmother on my maternal chromosome at 7 million, 5 million bases. That point is called a crossover. Wherever you see a change in these diagrams, where you change from one of your grandparents to the other, that's called a crossover. And you'll note that crossovers only happen to one sibling at the time and only one of their two chromosomes. So what we have to do to get started in the process is to compare the four siblings with each other. If you have four siblings, there are 6 comparisons to make. Sibling 1 with sibling 2, sibling 1 with sibling 3, sibling 1 with sibling 4, sibling 2 with 3, 2 with 4, and 3 with 4, 6 of them. And we need diagrams for these. Now, first I'm going to simplify things a bit, and we're going to consider a family of 3 siblings. And this method was first suggested by Kathy Johnston, and the article that I'm going to provide to you is co-authored by Kathy and myself. So if you have 3 siblings, then there are only 3 comparisons to make. Sibling 1 with 2, 1 with 3, and 2 with 3. Someone was asking just before the meeting started about a site where you can upload your data, and this is it here. It's called gedmatch.com. So you can find that easily on the web. So here we have the 3 comparisons between the 3 siblings. These siblings are labeled B, K, and W. I think the K is actually Kathy Johnston. Where there's a dark blue band at the bottom of the comparison diagram, that means that's a half identical match. Where there's gray or brown at the bottom, that means no match. And where there's green just above, that means a fully identical match. As I said, among siblings, you will see some fully identical segments. But every place where the type of match changes, like from fully identical to half identical at this point right here, then that represents a crossover, and that crossover belongs to one of the people involved in the comparison. In this case, it happens to be B's crossover and not K's. Let's go back one and look at this again. So this just identifies the crossovers of each of the 3 siblings. All the ones with the arrows in the top represent B's crossovers. The middle set represents K's, and the bottom set represents W. B has 4 crossovers. K has 2 here and here, and W has 2. But we don't know on which of their chromosomes we have 2 copies involved. We don't know whether it's the paternal side or the maternal side where the crossover occurred. That's one of the problems to figure out. So just looking at these 3 comparison diagrams again, I've put vertical lines in marking all of the crossovers. And again, we have 4 for B, 2 for K, and 2 for W. Down at the bottom, I have indicated what kinds of matches are occurring in each of the segments between pairs of crossovers. For example, I'm scoring a fully identical match as a 2, that's for 2 chromosomes, a half identical match as 1, and no match as a 0. You see, on these 2 comparisons, we have no matches. So that's 2 zeros down here. And we have the fully identical match in the top diagram, and that's indicated with the 2. Now, as you go from one of these regions to the next, whoever owns that crossover, B in this case, any of the comparisons involving B, which would be the first and the third, those 2 match types have to change. In this case, it goes from a double match to a single match with K versus B, and no match to a half match or a single match looking at B versus W. Since K was not involved in this at all, and that particular match type doesn't change as you go across that crossover. So here I've labeled all of the different matching schemes for each of these segments. Now, what we want to do is color code each of the 3 siblings' chromosomes. A good place to start is where you have a double match, and that's what I'm doing here. We know that B and K are fully identical in this segment. So we draw in BK's chromosomes and just assign colors arbitrarily. We'll assign purple to one of their chromosomes, green to the other. We don't know if purple is on the paternal side or maternal side. We have to figure that out later. But we also know that B is not matching at all to K and W. I'm sorry, that W has to not match either of the other 2. You see, these bottom 2 diagrams are comparisons involving W. And we see that there is no match to either in this region. So we color these 2 segments entirely differently. Sorry. Now, the chromosome's source does not change until you get to a crossover. For example, K does not have another crossover till you get out to 72 million. The locations are down here at the bottom. The last crossover is at 72 million. So we can extend K's chromosomes colored just the same all the way out to that next K crossover. Similarly, we know that K and W continue to be totally different all the way out to the end. K doesn't have another crossover at all. I mean, W doesn't have another crossover all the way out to the end. So we can extend those colors all the way out to the end. We also look back to the left. We can extend each of these until we encounter another crossover. So we can go one more segment with W till we reach a W crossover, and we can go two more segments till we reach another B crossover. And out here on the last segment, we know that W and B have to be identical. So not only do we have W's coloring figured out, but we know that B has to match completely out here. So we've already filled in a amount of the chromosome, the recombination diagrams for this family of siblings. But now we're stuck because we can't really extend these any further without making some assumptions. But you get one free guess, so to speak, because we don't know which of these, we don't know if purple is on the paternal side or maternal side, but we can just make an arbitrary assignment at this point. We could say, and we won't lose any generality here, we can just say at this point, it's K's green chromosome that changes. And so we can change the green to the other possibility, which is gray. So since there's not another crossover for K, then we extend the green not only past this crossover, all the way back to the beginning. So we've got a whole lot of K's diagram done. And we know that B and K are identical in these two segments. So we can fill in B's coloring here. Now, at this point, we're kind of stuck again because, well, I'm sorry, we can go one step further by looking at the match types in these two areas. We know that K and W are completely different in these two segments, and so we can just pick the opposite colors in these two segments for W. Sorry, now we're stuck because we could fill in the top chromosome with orange and the bottom one with green. That would satisfy the matching perfectly well, but so the other choice. So we don't know which of these two that we have. And so really, we have two possible solutions from this point. And actually we have two more down here. So this is about as far as you can go just on matching information and logic. From this point on, you're going to have to depend on matches with cousins where you know what the relationship to you is. That is, which of the which of your four grandparents these cousins are related to you through. For example, both B and K match a third cousin who is only related to the siblings through the paternal grandfather. And it's a nice, so these two diagrams up here are with the matches between B and K. I'm sorry, between the B and the cousin and K and the cousin. top one is B versus the cousin, the bottom one is K versus the cousin. Now, notice that the upper chromosome changes from orange to purple in the middle. So you couldn't have a match that goes across that change, but the green is continuous in both B and K, and it's different in W. And so that fits the match information exactly. The cousin matches B, the cousin matches K, and the W does not match that cousin. That tells us immediately that the green, wherever it shows up here, not just right here, but wherever it shows up, came from the paternal grandfather. Then we find another cousin match. This one matches all three cousins. The match diagram is down at the bottom. It matches all three out at the end, and it matches W over a fairly large region. So that means that, oh wait, one other thing I should tell you. Once you know that the green came from the paternal grandfather, that means that the other choice on that chromosome, the gray, has got to be from the paternal grandmother. That's the only place it could come from, since this is all from one parent, came through one parent, and has to be from one or the other of the grandparents. So that means we can fill in, we can identify this last segment, and we can fill in K's segment where, because K matches this cousin over that particular segment. Now, because W matches the cousin throughout this region, sorry, this now gives you the solution that we were looking up here. Since this is this involves a crossover, B has a crossover in this chromosome. At this point, it has to change to I'm sorry, I'm getting myself confused. The gray well, first this shows that this gray part comes from the paternal grandmother, but it also tells us that in this segment, the orange is continuous because B stops matching this cousin right at this crossover. So that means that B changes at that point from gray to green. And then that determines the other color as well, because orange and green were the possible solutions to this part. And now, one additional thing that we need to, that we can, in this case, just they had an uncle, these siblings had an uncle who matched them, and you could use his information to determine that B's first crossover, this crossover to B is on the paternal side. And that gives you the key that first segment has to be shaded gray, this little tiny piece here. Next, we have to go and find cousins on the maternal side. And this was done, and I won't go into all the details because we're kind of running short of time. But finally, we have this complete diagram of how recombination happened in these three siblings. And if there's a match to any arbitrary unknown person in the database, by looking at these diagrams, recombination diagrams, we can tell exactly which grandparent that the match to the unknown person is through. And that's the value of this. Now, this same approach will work for any number of siblings, as long as it's 3 or more. The more siblings you have, the fewer problems and ambiguities you'll have occurring, but also the logic becomes a bit more complicated. We don't have time for a full discussion of the numerical approach, but this article is available at this particular URL. There's also an Excel spreadsheet that will allow you to use the method more or less automatically. And that Excel spreadsheet is also downloadable. And there is another Word file there that has instructions on using the spreadsheet. But each of there's one spreadsheet with 4 strings. If you change that 4 to a 3 in the URL, you'll get the instructions and spreadsheet for the 3 sibling case. And the PowerPoint presentation that I've just gone through is also available, and there's the URL for downloading that one. So this approach can completely resolve how recombination occurred, as long as you have the comparisons between siblings and you have a few cousin matches. So I guess we'll stop at that point, and I'll be happy to answer any questions.
Well, thank you, Whit. That's for a very, very interesting presentation. I guess I was thinking about it as you're going through, and unfortunately, I only have one brother, so we'll have to figure out some other way to track down our grandparents, I guess. But
you can sometimes do this with just 2 siblings if you have a lot of known cousin match to compare to.
This reminds me of on Ancestry, where you have an option to identify a DNA match from your list, your match list. Do you know who this person is? You could say they're a second cousin once removed, or because you have figured out who they are. Is that helping Ancestry's algorithm to be more accurate?
I think that it is for you. I'm not sure they're using, I mean, they may be using some statistics out of this to fine tune their estimates as to what the relationship is. But I think it's mainly valuable in your own matches to other people. If they know for sure how you're related to several people in the database, it helps them to figure out how you might be related to people that you don't know.
Can we
Okay,
use
thanks.
DNA Painter to accomplish this?
Joel and I were having a discussion about this before we started. I have not used that DNA Painter toolkit. It's on my list of things I want to do. The DNA Painter will go beyond grandparents, but if you have this combination done on your back to grandparents, it will help you with the subsequent generations going back. But like I said, I have not used those yet, but yes, they should give you the same results.
And then on My Heritage, they have something? that
I
groups people, so that supposedly everybody in that particular group everyone has a common ancestor, the same common ancestor?
Well, if you have a group of people who have a half identical match at the same location or the segments overlap, then it's very probable, highly probable, that all of that DNA in the part that overlaps came from the same common ancestor. Yes. They call that triangulation in
Right,
some circles.
right.
Joe,
Yes,
will you post the links that were shown?
yes, I will. I'll, we can get a copy of the PowerPoint too, but I'll send out an email with all of those links.
Thank you.
I'll provide the Joel.
We have a question from Perry who's asking, can this method help us figure out how a DNA match to a third cousin is related to us? And I think the answer is yes.
Yes, it is. Probably not a lot of people know who their third cousins are, but if you know which grandparent the relationship goes through, it knows the focus considerably. It helps you figure it out.
So yeah, I guess this means I'm going to have to go back into my emails from MyHeritage and start figuring out all the links that they send me.
Yeah.
Okay, so if there aren't questions directly on this topic, does anybody else have any comments or questions more generally about genetic genealogy?
This is Chuck McKean. I'll just make a comment. Another thing maybe that would be of interest that is sort of similarly related is some work that Amy Williams at Cornell did. And she has this tool called Happy, and from it's a computer program, but from 3 or more siblings, you can, and a parent, through the automatic phasing techniques, you can identify the grandmother's DNA from parents from 3 siblings and 1 other parent, because it sort of does this phasing automatically. Figure out where it's changed.
Interesting.
I'm sorry you were breaking up a bit, so I couldn't catch all of that. But I know that there are people out there who have tried to create tools that would implement this automatically. Unfortunately, there's a lot of manual work that has to be done, because the crossovers are sometimes hard to locate. If you have, this can happen both when just accidentally a crossover occurs in 2 siblings that are very close together, but also the endpoints of the matching segments are a bit fuzzy, and you won't find exactly the same endpoints in the different siblings match data. So you have to do a little bit of manual massaging of the data to sort of harmonize the endpoints in order to get any of the automatic tools to work. The visual method has the advantage that you can do that just by eye, usually, and you can just figure it out from the diagrams and from logic. someone asked the question about the situation of a half cousin. Well, if you have, say, a half first cousin would have roughly half of the DNA match with you as a full first cousin would have. The same with a half sibling. Could also throw a little cautionary note in if you're just considering having your autosomal DNA tested, you will get, after being tested, a list of people you're related to. And some of these relations will be close cousins to you, and some will be distant. But the sort of unsettling thing that can happen sometimes is that you will find in your list of relatives a half sibling that you didn't know about. And that's sometimes upsetting to people. Other times it just brings up really interesting stories.
I don't want to go down another potential rabbit hole, but I know that that Whit, you're the administrator of a number of Y chromosome projects, and several number of the people in the group have a significant interest in understanding Y chromosomes and also maternal mitochondrial DNA from our mothers. Do you have any general comments about that?
The mitochondrial DNA is a bit of a problem because you only get a mutation in a particular line about every 3,000 or 4,000 years. So if you have an exact match to someone, you do have a common matrilineal ancestor, but that ancestor could have lived in the last century or thousands of years ago. So it doesn't really help you to look through a match list of mitochondrial DNA. But what you can do, and what I have done myself, is you can prove or disprove hypotheses. If you are working with a distant cousin that you are related to along a strictly matrilineal line, and you would like to know if you and your cousin have got your genealogy correctly worked out, well, do the mitochondrial testing. If the only way your genealogy can be correct, that you're related through strictly matrilineal lines, is if you have the same mitochondrial DNA. And if you don't, you've just disproved your hypothesis. Now, on the Y side, the Norwegian community has a bit of a problem. Most of these Y chromosome projects are also called surname projects because they depend on the fact that people with the same surname have passed down not only their Y chromosome, but their name as well, the surname. There are places like in Wales where they only got around to adopting surnames, a standardized way of passing them down in the middle of the 1800s. So I have one Y chromosome project for the surname Owen, and we have over 100 independent Owen Y lines. As for my own surname, Athey, just about all the Atheys are descended from the same immigrant ancestor, and they all dutifully pass the name down a strictly male line. So it's in the Scandinavian countries, you have different surname conventions, and it makes it more difficult to follow a name or a Y line.
Yeah,
it's sort of similar. It's similar to the mitochondrial case, because if you look back a matrilineal line using standard conventional English naming systems, you still have a surname change at every generation. And so Scandinavians, it's often more like that, even on the Y lines.
Is an X match useful at all?
Well, I think it also is, it can be useful for disproving hypotheses because of the fact that you never have an X transmission between males. Males pass only a Y to their sons, they pass an X to their daughters. So if you have a hypothetical genealogical line that you're trying to prove, and you have an X match, and there are two consecutive males in your line that you're trying to prove, then something is wrong because that didn't happen. And just ordinary X matches can also be useful. It's in females, they do recombination between their two X's, just like all of the other chromosomes. But in males, there's nothing to recombine with because you have one X and one Y, and you can't do much recombination there. Actually, there is a little teeny tiny piece on the end of the Y chromosome that does recombine sometimes with an X, but that's kind of rare. I mean, it's not rare, but it's not useful.
Okay, I'm going to stop recording.